Hello, good morning students. Myself, Dr. Balasubramaniam, working as Assistant Professor in the Department of Zoology, Government First Grade College and PG Center, Chintamani. And today we will discuss about the life cycle of unio. And life cycle of unio, it is a part of the type study of unio. And this topic is belong to the second semester basic zoology syllabus. So last class we saw about something about the reproductive system in the case of the unio, and the reproductive system in the case of the unio. Here these males possess the testes while the female possesses the ovary, and this unio is said to be dioecious and sexes are separate, but there is no sexual dimorphism. Externally we cannot distinguish which is a male and which is a female unio, but sexes are separate, and therefore it is dioecious. Now this dioecious also you can call it sexes are unisexual, sexes separate, unisexual. Next, the gonads testes in the males and ovaries in the females. Next point, the gonads are paired, large, highly branched structures lying in and around the intestinal coils in the visceral mass above the foot. Now this is characters. During the breeding seasons, they become greatly enlarged and conspicuous, and here the testes are brightly whitish in color and ovaries are reddish in color. Now, as we know, the testis has the germinal epithelium or the pyramidal germ cells through which there is a spermatogenesis process happening, and finally the spermatozoa, the sperms are obtained. Similarly, the ovary also has the germinal epithelium or the pyramidal germ cells through which the oogenesis process happens, and that phase of this multiplication and phase of growth and finally the phase of maturation leads to the formation of the eggs or the ovum. Now, in the life cycle of unio, last class we discussed till the formation of zygote. Now what happens here in the case of the unio? The organisms, the female release the eggs from the ovary, and these eggs doesn't come out from the body of the unio, stay in the water tubes of the gill lamina, and here these places are called the brood pouches or the marsupia, and here by chance these eggs are met with the sperms and the zygote is formed, and this through the water the sperms are carried through the inlet siphon, and they meet the egg, that is the ovum, and the fusion of the zygote is formed. Now the fertilization is species specific, and it takes place by chemotaxis, and the sperms as the anti-fertilizin and ovum as a fertilizin, and this egg as the gynogamone and sperm as the androgamone, and because of this species specificity there is the formation of the attraction, and these sperms fuse with the eggs to form the zygote. Till that stage we saw the zygote formation. Now the zygote undergoes complete but unequal division, unequal cleavage, resulting in the hollow ball of cells called blastula. So what is cleavage? Now cleavage is the rapid repeated mitotic division of the zygote. Cleavage is the rapid repeated mitotic division of the zygote. Now this cleavage happens resulting in the hollow ball of cells called the blastula. Now the blastula consists of two types of cells, namely the smaller, clear, yolkless and rapidly multiplying macromeres on the upper side, while the large yolk-filled and slowly dividing macromeres at the lower side. So two types of cells are there in the case of the blastula, and the small cells are called the micromeres, while the large cells are called the macromeres. Now this is the division occurring in the case of the zygote of the unio. Further, the invagination of the micromeres on the lower side into the micromeres results in the gastrula. So there is a process of gastrulation. Now we know that in the gastrulation, so three germ layers are formed, And this gastrulation is brought about by the morphogenetic movements of the blastula. And blastula is formed, micrometer cells in the gastrula, and the gastrula bears a tuft of cilia posterior. Some of the cells are budded off into the blastocele to form the mesoderm. Now here is a very important step. In the case of the gastrulation, we have the invagination, we have the involution, we have the emboli, divergence, convergence. A lot of changes are taking place in the gastrulation, and this gastrulation is called morphogenetic movements, and this gastrulation brings about the 3 germ layers. So therefore this gastrulation is a very important step as far as the development of any organism is concerned. So the invagination of the micromeres on the lower side into the macromeres results in the gastrula bearing the tuft of cilia posteriorly. Some of the cells are budded off into the blastocele to form the mesoderm. So mesoderm is formed, as well as the gastrocele or the archenteron is also formed during the development. Simultaneously here, a deep invagination on the posterior dorsal side of the embryo forms a characteristic molluscan organ, the shell gland. So this is peculiar type in the development of the unio, that molluscan organ is formed, that shell gland secreting first an unpaired embryonic shell, which is replaced later by the bivalve shell. So over this process gastrulation, during the gastrulation process, so there is invagination, involution, all these morphologic movements lead to the formation of the molluscan organ and that shell gland secreting the unpaired embryonic shell, which is replaced by the later bivalve shell. This further, the valves of the shell are triangular in nature, and the ventral angles of each triangles bears a serrated hook. An adductor muscle is also formed by the mesoderm shell. So we find the hook formation and find the adductor muscle, and we find the mesoderm shell formation. The same thing exactly, all these features are found in the glucidium larvae. So meant to say here there is the zygote formation, followed by the cleavage, followed by the blastula, followed by the gastrula, and gastrula leads to the glucidium larvae. Now the mouth of the gastrula closes, and definite mouth is subsequently formed by the invagination of the ectoderm. See how the mouth is formed by the invagination of the ectoderm. The mouth of the gastrula closes. A definite mouth is formed, subsequently formed by the invasion of ectoderm. So this is how the process of formation of mouth is formed. The body of the embryo splits from the ventral side to the dorsal side and gradually changes into the larvae called glucidium larvae, the characteristic larvae of freshwater mussel unio. So this is a very important step. So this embryo splits from the ventral to the dorsal side and gradually changes into the larvae called glucidium larvae. That is a characteristic feature of the freshwater mussel. Now, next further characteristics. The glucidium larvae means point of arrow. So it is a pointed structure. It is a minute larvae measuring about 0.1 to 0.4 mm width. The shell consists of two triangular and porous valves united dorsally but free ventrally. The ventral free end of each valve is produced with the conspicuous hook beset with the many spines. So between this hook and valve we find many spines. The shell encloses the body with the right and left mantle lobes. So inside the shell we also find the mantle lobes. Now, in the glucidium larvae, the foot is not yet developed completely, but mid-ventral surface of the body as a glandular pouch which secretes the long sticky thread or the larval byssus. Now, tremendous number of glucidium are found in the outer gill lamina. Now here, these glucidium larvae are found in the outer gill lamina. Now we observe that the egg is deposited in the water tubes of the gill lamina. From the eggs till the formation of glucidium larvae, that step, that organism, that is glucidium larvae, stays in the gill lamina itself. Inside the glucidium larvae remains entangled by them, by their bases, and nourish the mucus secreted by the gills. So here the bases nourish on the mucus secreted by the gills. So here there is a glucidium larvae stays in the gill lamina of the female unio. Now further, when sufficiently mature, they expelled, that is, the glucidium larvae are expelled through the exhalent siphon into the surrounding water, where they slowly sink to the bottom or scattered due to the water currents. So this is what happened. So here the glucidium larvae is expelled through the exhalent siphon into the surrounding water, where these larvae either slowly sink into the bottom or scattered due to the water currents. Further, what happened? Some of the larvae attached to the skin or fins or gills of the fish by closing their hooked valves and penetrating the host tissues by the byssus thread. So here the hooks are there. These hooks helpful in the attachment to the skin, fins or gills of the fish, and this byssus thread penetrate the host tissues for the extraction of the nutrition. Now in this condition they lead to a truly lead, that is, the glucidium larvae lead a truly ectoparasite life, receiving nourishment by host by absorption for about 10 weeks, during which they undergo peculiar metamorphosis to the adult. That is, the glucidium larvae initially attached to the gills, skin or fins of the fish, and now here it takes around 10 weeks where it metamorphoses to the young adults. Now during metamorphosis, what are the changes which happen? We see in the next slide. Now during metamorphosis, the larval structures such as sensory bristles and byssus thread disappear. This is what happening during metamorphosis, where at the surface of the skin, gills or fins of the fish. So during metamorphosis, the larvae structures such as sensory bristles or byssus disappear and single adductor replaced, single adductor muscle replaced by two adductor muscles. The mantle is replaced and ectoderm lying posterior end of the enteroderm ruptures to form the anus. So here single adductor muscle is replaced into two adductor muscles. The mantle is replaced and the ectoderm lying posterior end of the enteron refer to the digestive canal. Now this digestive canal or archenteron or enteron or gastrocele ruptures to form the anus, that is, the anus is formed towards the posterior side. First the mouth was formed and now the anus is also formed. So it is complete digestive system. Now foot makes its appearance. Now till now the foot was not there. The foot now makes appearance. Gills arises, ctenidae arises, and the rudiments of visceral organs make their appearance. So here the rudiments of visceral organs make their appearance. That means it may be the digestive system, it may be the excretory system, maybe the metanephilia, it may be the reproductive system, it may be the coelom, it may be the digestive gland, it may be the gonads, etc. All the found in the visceral organs, they make their appearance. After metamorphosis completely, the cyst walls weakens and breaks, liberating the immature muscles, the freshwater muscles that fall to the bottom. Here, they feed in the normal adult manner and gradually acquire the adult shape and size. So, finally, what is the significance of glucidium larvae? The life cycle of parasitic glucidium larvae on fish hosts has many advantages, like it gets protection from the enemies or predator because attached to the surface of the gills of fin or skin of the fishes, so usually it cannot be eaten by the predators. So therefore this glucidium larvae get protection. Second thing, it get nourishment. Now here there is a byssus thread which penetrates into the host tissues, extract the nourishment, and this larvae grows. That is the second significance of the glucidium larvae. Third is the rapid dispersal and proper distribution. So here the unio is a sluggish animal, slow moving animal. It is found in a particular region. So therefore this glucidium larvae, whenever attached to the fish, so the fish keeps on moving to the far off places, and therefore there is a proper distribution of the animal, and survival of the unio also very important, because the animal is a sedentary, sluggish, slow moving animal. Without this dispersible, there would be competition for food resources in a particular region. So therefore to reduce the competition of the food resources, so this formation of glucidium larvae becomes very much important. So this is the diagram, very important diagram of glucidium larvae. So where we observe the byssus thread, we find the hooks, we find the sensory bristles. So this is a byssus thread for nourishment, hooks for attachment, sensory bristles for tactile in function, sensory to function, mantle is there, shell valve, adductor muscle, single adductor muscle, and hinge is there. So each and every things are here very particularly useful for the leading the ectoparasitic life. So this is the life cycle of unio. See what's happening. So this is a nutshell. In the nutshell we have all this slide showing here the male unio, freshwater mussel, release the sperms through the excellent siphon. Now this excellent siphon reaches the female through the water currents, and in the female there are the gill water tubes in the gill lamina, and here this eggs are there, and this fusion of the sperms and ovum will take place, and it forms the zygote, and the zygote later on forms the cleavage, and this cleavage later on forms the blastula, then forms the gastrula, and here this development takes place. So that's what this brood sac, that is the outer gill lamina, contain the developing eggs. So here once this completely matured, that's the glucidium larvae is matured. Now glucidium larvae is released through the excellent siphon, and some of the glucidium larvae sink in the bottom, and some of them move in the water currents, and by chance some of the glucidium larvae get attached to the gills of the fishes, fins of the fishes, skin of the fishes. So this is the glucidium larvae discharge, and here the attachment to the fins, gills or skin of the fishes. So further what happens, there is a formation of cyst around the glucidium. So here there is a metamorphosis taking place, and once this metamorphosis is completed, now during metamorphosis we know that the byssus thread is reduced, and the spines are reduced, and single mantle is replaced by single adductor muscle replaced by two adductor muscles, and the mantle is well developed, and the tilde are developed, gills are developed, the visceral mass rudiments are developed, and in the visceral masses a lot of organs are developed in the visceral masses. Like that, so much metamorphosis takes place, and finally the cyst weakens, and this glucidium falls into mature, and here these young ones fall in the soil, and they gradually develop the young unio, and later on the same unio become the adult unio, that is a male and female. That's how the life cycle goes on continue. So this diagram is very important as it shows the entire thing in the life cycle of unio. So again this diagram as far as the short notes concerned, glucidium larvae. In the second semester we have a lot of larvae are there, particularly this larvae and echinoderm larvae and Paralleloclastis tornaria larvae. So many things are there. Okay, summary. So the embryo splits from the ventral to the dorsal, and gradually this changes to the glucidium larvae, the characteristics of the freshwater mussel. So finally references we saw, that is the Code Part textbook, and Jordan Verma and Kavanataya, Damya and Esen. So out of this Code Part textbook is a very good textbook as far as the topics freshwater mussel type study is concerned. Thank you for listening.